Hello, everyone. My name is Peter Sagona, and I work with SiO2 Materials Science. I head up the applications group and been with the company since it was founded about 10 years ago. I'm pinch hitting today for my colleague, Chris Weichert, our chief scientist. And the talk today is on our next generation vials for specifically for COVID-19 vaccines, cell and gene therapies. And look forward to speaking with you about that. And let's get right into it. So some of the challenges with the COVID-19 vaccines and gene therapy drugs that are emerging drugs that are being developed, all of them are biologic based, and those drugs tend to be complex molecules, and they're inherently unstable in liquid formulations. And so those are some of the challenges we've seen. And many of the COVID-19 vaccines today require cryogenic storage to meet the shelf life. And there are efforts ongoing to try to lyophilize these drugs to eliminate the cold storage requirement. So these are some of the things. And let's kind of get into what SiO2 offers. So SiO2 is a manufacturer. Our plant is located in Auburn, Alabama, about 100 miles from the Atlanta airport. And what we do is we make primary containers that combine the best attributes of glass and plastic and none of the drawbacks. So we call them a hybrid parenteral vial. Hybrid because they're a combination of glass and plastic. Materials of construction, there are two components. The primary container is made of a polymer. And in the case of the vial, we injection stretch blow mold it. Injection stretch is similar to injection blow molding, but you have a smaller stretch ratio, and that helps us control the wall thickness of the vials. We use medical grade resins, and we focus on cyclic olefins, that's cyclic olefin polymer, and cyclic olefin copolymer, COP and COC. These polymers have, with molding with them, very good dimensional stability, very good consistency so we can manage and keep tight tolerances. The containers are optically clear, and I think the important thing is that they don't break. Also, cyclic olefins have a fairly high barrier to water vapor, so they keep water out or in, depending on the application. And then on this container, in a separate process, using plasma enhanced vapor deposition, we deposit a very thin glass-like coating. It's silicon-based. It provides enhanced barrier for atmospheric gases, in particular oxygen. And another important feature is that it's a clean, inert environment where there are no extractables, and then leachable studies with drug product have shown little or low leachables. It also provides barrier properties to prevent inks or label adhesives that would be applied onto the outside of the vial from migrating into the drug product. So the barrier system actually blocks materials from anything that could be on the outside of the vial, as well as atmospheric gases. And this glass coating, when it is applied to the optically clear vial, retains the optical clarity. The advantage of the coating technology is it can be applied to any container geometry. SiO2 Materials Science manufactures a line of vials as well as pre-filled syringes. The chemistry is identical. We have, and plasma enhanced vapor deposition is a process that really involves three elements. It involves a vacuum, applying a vacuum to the vessel that you want to coat, and then delivering a mixture of gases into the container, and then an electric field to actually excite the gases into the plasma state. Now, plasma enhanced chemical vapor deposition is a common use of applying coatings. It's used in many other applications, and it's generally chamber-based, where you put hundreds or thousands of parts in a chamber, you pump it down into a vacuum condition, and then apply the gas, which can then apply the coating. In this case, we use the container as the vacuum chamber. We coat them one by one. And what you can see here is a coating system for vials as well as pre-filled syringes. Coating them one by one gives us very good uniformity of both the thickness and as well the chemistry. And this coating system that we have, that I just presented to you, has been scaled to an industrial level where we're able to manufacture upwards of 10 million vials per month. Now, the barrier coating system is really a series of layers. It's what we call our tri-layer, but the overall thickness of this barrier coating is less than half a micron, so very, very thin. And that's a very important characteristic, because a plastic vial can deflect if you bend it. And so there was always a question of, hey, what happens if you deflect or bend the container? Doesn't it crack the coating? Well, a key point of this is being able to get a functional barrier that's very thin. And so at half a micron, it's a very functional barrier, but incredibly robust to mechanical challenges. We have an adhesion layer, which is the layer that touches the plastic, which binds the stack to the polymer container. The functional barrier is the green part in the center. That's pure SiO2, and that creates the gas barrier and the barrier against leachates. And then the final one, which is the drug contact surface, is a pH protective layer that allows the container to support a wide range of pHs, from a low of pH of 3 up to a pH of 14. And the important thing also is that the drug contact surface is hydrophobic, unlike type 1 borosilicate glass, where it is generally hydrophilic. Well, next we'll talk about our lyophilized drugs and some of the properties of our containers and how they perform in freeze drying. So just at a high level, some of the advantages that our vials offer for lyo. Certainly breakage, or the lack of breakage. There is no concern about breakage due to dropping, thermal cycling, or that type of thing. We've also found that we have lower subvisible particles, particles that could shear, be created during shear forces that are created in the containers. We will show you some data that compares subvisible particles after freeze drying in our container versus a glass container. And another important one is drying consistency. Because we have very uniform wall thickness, tight tolerances, and certainly the mass consistency, the weight-to-weight variation of our containers are much tighter than glass containers. In the lyophilization process, we have a very consistent drying rate of the material. We'll highlight some of those features as we continue on. The hydrophobic. Yes. So again, one of the things in lyo is cell wall, or excuse me, hang up on the wall of the container, residual material that gets lyophilized. Because we have a hydrophobic surface, the liquid drains down into the bottom of the container, and you get a lyo cake without a lot of fogging or wall hang up. Also, another thing we found is less protein aggregation, and we have a case study to show you on that. And then another important one, particularly with multi-dose vials, which have been predominantly used now for COVID-19, the ability to lyophilize larger volumes in the container. We have done a breakage study showing virtually no breakage in the SiO2 containers at 100% fill volume. I think one of the things that we noticed, and you might be thinking, is, hey, what about the lyo cycle, particularly when heat transfer coefficient of plastic is much lower than glass. You can see an order of magnitude difference between cyclic olefin, COP, COC versus glass. But the important thing here is the heat transfer coefficient. The heat transfer coefficient is only one aspect. It's really the heat transfer is really driven by the coefficient and the thickness of the container, or so variation in thickness also affect the heat transfer in the container. So we've done a couple of things with respect to our vial to improve heat transfer. We certainly have inherent to the design is the sidewall consistency, the wall-to-wall consistency. But another thing we've done is, because we have we mold out of plastic, we've made flat bottom containers that increase the conductive heat transfer from the container to the shelf. And so this has made this, in combination with the wall thickness consistency, has really improved heat transfer overall. And so as you see here, here is just another measure of our consistency, both on the left, the mass variability, and you can see that we're more than an order of magnitude more consistent in less mass variability compared to glass. And then also the dimensional variation of and we're measuring the outside diameter. And so these two combinations make the heat transfer much more consistent with the SiO2 container. And as you can see, let me just go back to this slide here. So this is a study we did of doing a freeze-dry study of heat transfer, and the blue ones are where the locations within the tray. And here you can see that the heat transfer rate is much closer to glass than the original coefficient, the heat transfer coefficient, and that's due to the dimensional consistency of our container. And then you can also see that by the standard deviation. And I think this is an important point. And the folks at Milwaukee had conducted some lyophilization studies, actual cycle studies, and really the cycle time of that can be achieved with an SiO2 container is very similar to glass. Again, another thing that another feature, because of the consistency of the container, is the lack of the consistency of how the product dries, the primary drying, because that also has an effect on the cycle. And so we have a much more consistent drying. We've looked at the water loss after 18 hours and 23 hours, comparing the SiO2 flat bottom container against glass. And you can see that the amount of water loss is much more consistent with SiO2 versus glass. Moving on to breakage. This was a study that Milwaukee conducted, Milwaukee Technology, where we looked at vials with 10% mannitol, and they were put onto freezing shelves at -40°C, and then at a cooling rate of 1 C per minute, then held for 2 hours at freezing. And then the pressure was reduced to 150 millitorr, and then the shelf was warmed to 5°C. And again, what you can see here is that you start seeing glass breakage when the fill volume exceeds 50%. At 40%, there's no breakage, and then as the fill volume increases up to 100, you see more breakage. And that's not the case with SiO2, which is really, you get a lot more flexibility and with respect to fill volume. Another area that we studied was generation of subvisible particles after lyophilization. And here we are measuring particles in a 10% mannitol solution filled in a 6 ml vial. And what you can see here is a significant increase in subvisible particles greater than 2 micron after lyophilization in a glass vial, and virtually no difference in either in an uncoated or standard COP container and a SiO2 or silica coated COP container. We did a practical one we did a case study with a drug, an IVIG drug formulation, and we repeated this lyophilization study measuring particles greater than 2 micron. And again, here it's a side-by-side comparison of the SiO2 vial and the glass vial. And you can see that much lower subvisible particles, also and looking at the lyo cakes, the lyo cakes looked very similar in and also had a similar moisture content, similar appearance, but as far as subvisible particles after reconstitution, much lower in the SiO2 container. Again, as I mentioned before, because SiO2 containers have a hydrophobic drug contact surface, there is very little hang-up of the liquid during freeze drying, and so you don't have any residue that gets freeze dried on the side wall. You can see a comparison of a study that was done. It was a placebo with hemophilia, high salt content, and so it was one that it was prone to hang to the wall in a hydrophilic environment. So those are some of the things that have been done in the freeze drying area. I think from our perspective, I think it really provides an ideal container for doing freeze dried development and because of the consistency of the container and certainly no breakage. Next, I want to switch to cold storage because this really has been something of interest for COVID-19 vaccines and cell and gene therapy. One of the things that is required is really having good seal integrity of the vial at temperatures down to as low as minus 190 to 96 degrees C. We want containers with low particles, no leachates, so no extractables from the primary container, a barrier to oxygen, no breakage. Another thing that SiO2 offers with its container is we have a unique ID that can be placed on each and every container. It's not a batch ID, it's a unique container ID. That ID is used to capture information about how the container was made within SiO2, but that serialization, that ID is also available to be used by end users. And then finally, SiO2 vials are delivered in a ready-to-use format, in a nested tub or in a bulk tray. Both are in ready-to-use format. So one of the studies we did was a freeze-thaw study at minus 20 and at minus 70 degrees C. And the idea here was really to assess robustness of the container compared to glass. We took a group of 10 ml vials filled with 6.5 milliliters of fill volume of liquid, and then cycled them 24 hours, cycled them to minus 20 and minus 70 respectively, held them for 24 hours, then room temperature for 24 hours, and repeated that cycle 3 times for both the minus 20 case and the minus 70 C case. And what we found was we ran both 2 types of SiO2 containers, one with cyclic olefin copolymer, one with cyclic olefin polymer, and then 2 glass containers. All of the containers, glass and plastic, no breakage was seen at the minus 20 degree cycling. At minus 70 degree, you see that there was breakage even after 1 cycle in some cases at the minus 70 cycling. For SiO2, both types of cyclic olefin polymer type containers, no breakage was observed after 3 cycles. One of the other things that we've studied was container closure integrity, and I'm going to show you data both at minus 80 degrees C and then at liquid nitrogen at a lower temperature. In this case, we're using carbon dioxide as the tracer gas, the gas to identify leak. So we fill and we stopper the container. We capture atmospheric gas within the container. So that's why you have a mix of nitrogen and oxygen, atmospheric gas. We then put it into a chamber filled with carbon dioxide. That provides the driving force of gas that could permeate into the container. And then we lower the temperature to -80 and hold it at that temperature. And we're measuring, it's a non-destructive test where we can measure the change in concentration of CO2 within the container over time. And if you see the ratio of carbon dioxide going up in the container, that's a function of leakage. We ran a series of, we did a study with 10 ml vials using a series of stoppers. And these are the studies that we did. And it's a study that goes out over 12 months. We also created a positive control by intentionally drilling a 5-micron hole into the container. And again, since the analyzer was from Lighthouse Instruments. And right now we've got 6 months of a 12-month study. And what we're showing here on the left is the positive control, showing with the 5-micron hole in the container how the concentration of CO2 changes in the container over time. And then the test samples, which are using different stoppers, different types of stoppers. And what you can see here is very good container closure integrity maintained at -80 degrees C. For the next study, we're doing an evaluation of CCIT, container closure integrity testing at -180 degrees C. Here, we use a nitrogen gas because carbon dioxide would be a solid at these temperatures. So a very similar test setup, except using the nitrogen as the tracer gas. And in this case, we've got 3 months data. On the left side is a positive control. And another thing important to measure is, what are we measuring here? Well, rather than measuring carbon dioxide into the container as we did before, in this case, what we're measuring is oxygen concentration into the container. So at the starting point, because if we go back here, we capture atmospheric gas. So the starting point is 20%, generally 20% oxygen in the atmosphere. And what we're measuring in the positive control is the change in oxygen concentration. So what's happening is the nitrogen, that is the gas surrounding the container in the chamber, is migrating into the container. And so you're getting a decrease in oxygen because you're getting an increase in nitrogen. In the case of the test sample, over 3 months, the oxygen level is holding at 20%, which is the initial oxygen that was in the container at the start of the test. This test, again, will continue on for up to 1 year. So what we have here, though, is a very robust container that doesn't break and also can maintain container closure integrity up to -180 degrees C. The last thing I'd like to show you is a case study that was done with an actual drug. It was this was very kind of a customer to share this data with us. It's a peptide-based viral vector formulation, and this is a study that was done at -80 degrees C. It was shelf life study. And it's a 3-year study, and we've got over 1 year's worth of data. They came to SiO2 because they wanted to, they had ion migration problems packing in borosilicate glass. As you know, borosilicate glass is a combination of SiO2 with metals such as boron and aluminum and iron. And under certain cases, those ions can migrate into the drug product. So this chart here is kind of complex. I mean, it's got a lot going on, but generally what this is showing, if you just look at the time along the top from t = 0 up to time equal to 12 months, and you just look at the attributes, the appearance, identity, and so forth, what you'll see is that really there is no change in the drug product based on these attributes over time. And what that is showing is a measure of stability of the drug product in the SiO2 container. And so this is one of the cases where the SiO2 container is one that performed very well at minus 80 degrees C and is really a suitable container for this application. So just to wrap up what we presented, when I know I've given a lot of material here, is we developed a container for freeze-drying lyophilized drugs and also for vaccine products. Lower heat transfer variability means lower freeze-drying and lower freeze-drying variability. We've shown no breakage and no wall residue, so very good, attractive-looking lyo cakes. And then also an important part is fewer particles and aggregates compared to glass after the freeze-drying cycle. And that was done with both a model solution as well as an IVIG drug. And then on the cold storage side, we've demonstrated that we can withstand very cold storage. Both we've demonstrated container closure integrity at minus 80 for 6 months and at minus 180 for 3 months. Also a case study with a viral vector formulation stored at minus 80 degrees C for 1 year with no degradation of the drug product. And again, here, we've also done freeze-thaw studies at 70 degrees C, showing that there is no breakage. And so you have a very robust container that we think is very well suited for applications that involve cold storage or lyophilization. So that's the end of the presentation, and I would be very happy to take any questions that you may have.